So here's the neuron. And in this model, these structures over here, so this is the cell body, these structures, the branches, that have been clipped off. We don't see the ends of them, right? There are these long tendrily spikes that will project out into whatever neural tissue. These are the dendrites. That's the incoming source of information, all right? And they impinge on the soma, the Greek word for body, or the cell body. And at the center of the soma, we see the nucleus. And I notice on here that I don't have synaptic bouton, but I should. It's this structure. These little like Hershey's Kisses with the little thing sticking off them. Those are synaptic boutons from upstream neurons. Those are from another neuron, right? So it's a different neuron that is synapsing on the dendrites in the cell body. So I said the nucleus. Surrounding the nucleus on that model, you'll see a little bit darker airbrushed paint there, which represents the perikaryon. I think this is the point. So I sort of preemptively tried to launch my joke about Rick Perry earlier in the semester. This would have been the right time to do it. The perikaryon is the cytoplasm. The neuroplasm is the actual term, the neuroplasm, that is directly surrounding the nucleus. And this is where, this actually fairly complicated cytoplasmic architecture is represented by some blue spray paint. But this is where you're going to have, for example, microtubule lattices that are being impacted by summative electric fields created by graded potential from synapses at all these synaptic photons. All right. Maybe, probably not, but maybe I'll go off script a little bit and talk about how electric fields affect quantum states and the microtubule lattices in there. If I don't, it's an interesting thing that maybe that comment will stimulate some of you guys to explore on your own. Axon hillock. So after we get a summation of all these graded potentials, we have local currents that bring the transient that bring the passive depolarization of the axon hillock to threshold. So this little lump here is the axon hillock. Then we have excitable membrane of the axon until we get down to the axon sheath along the length of it. And that myelin sheath is created by what's called a Schwann cell, SCHWANN. It's an eponym. And this is one Schwann cell here, right? And then this area is a node of Ron VA. So this is an internodal region, a node of Ron VA, another eponym. Then you will notice on the outside of that axon is, you see on the very tail of it, it would be down here. There is this this gray covering, that's sort of rough gray covering. This is meant to be a connective tissue sheath called the endoneurium. The endoneurium. My tongue to wake up fully here. The endoneurium. And endoneurium is completely analogous to the endomysium that we saw as the connective tissue sheath along a muscle fiber. All right? Along an individual muscle fiber. So underlying it is the neurolemma. The neurolemma. And the neurolemma is the actual, so neurolemma is this layer, the outermost layer of that's underneath the Schwann cell. Axolemma is the excitable membrane along the axon. Yeah. Mm-hmm. Yeah, so on this model, it would be any of this pink would be a neurolemma, this would be axolemma, and then the bits. Yeah. All right. So here we're zooming in quite a bit more to one of the synaptic boutons. Vicky, please model it for the class. That is a blow-up of one of the little Hershey's Kisses, of which there are about 40 on that axon in front of Haley. And the neuron in front of Haley. So let's look a little bit closer. This is part of a telodendrium. Inside, we're going to have mitochondria. Mitochondrion, which is the plural there? Mitochondria is plural, correct, yeah, and mitochondrion is the singular. So mitochondria are responsible for the production of ATP. And this whole process requires plenty of ATP, so there's going to be a mitochondrion on site in the synaptic bouton to help with the ATP, serve the ATP needs of the synapse. This is the presynaptic membrane down here. The plastic base that it's sitting on would be the postsynaptic membrane. That base has been lost to the winds of time, but in between would be the synaptic cleft. So you can call this whole thing down here, this structure I learned it as synaptic bouton in a lot of anatomy texts now. They are less fancy and just use the word knob, synaptic knob, a little bit more humble. And then we see synaptic vesicle. So these little white balls in here are synaptic vesicles that are full of neurotransmitters. That represented little yellow speckling in there. So that's pretty much what is important in that model. Okay, here's some histology. Astrocytes. So astrocytes are well named. They're aptly described by their name as star-shaped cells, the astrocyte, the star-shaped cell. Let me see them here. There is, Grace and Amanda have the stack of histology. Everybody's got scopes in their bench. You can find wherever it is, but astrocyte is out there somewhere. I presume they put one out. And astrocytes actually are not neurons. They are neuroglial cells. We will find out what that means in lecture today, but they are not conductive cells. They're not excitable cells. They are supporting cells, and we'll see what they do in lecture. But this is what they look like. They're really distinctive. They're easy to identify. They don't have a long axon sticking out of them because they're not conductive, but they are star-shaped, these star-shaped cells, all right, astrocytes. You don't have to know anything else about them in their morphology, at least, their anatomy, besides recognizing them. Okay, now on to a neuron, a multipolar neuron. A multipolar neuron is the majority of the neurons in the nervous system. And here's the cell body, or the soma. You can see the nucleus in the middle. And then there are dendrites and an axon that comes off of it. A single axon of multiple dendrites. Sometimes it can be hard to tell the difference. Usually the axon hillock is the largest projection off the cell body, but sometimes it's hard to distinguish. So for example, in this one I would guess this is the axon hillock, and then these here are dendrites that are coming off of that axon. Off of the soma. Multipolar neuron. You'll see those slides. Yeah.
How do you know the
well, what is a regular? I'm not sure.
So
This is a regular neuron.
Okay, so all neurons are.
Nope. Not even close.
Okay.
No, we'll talk about that in lecture today as well. Yeah, but I'm just showing you this. This is the regular neuron, and it's this is the typical anatomy of it. It's called multipolar because the cell body itself. Again, this is anticipating lecture, but the cell body has multiple poles coming in. There is multiple dendrites, and then there is an axon coming out. So there are multiple poles. There are unipolar neurons. There are anaxonic neurons. There's a bunch of different types, but those are more specialized in structure. This is a very standard. So that would be a multipolar neuron right there.
Yeah.
Okay. Here's another one. This one demonstrating how difficult it can be sometimes to actually identify the axon hillock. If on a slide I were to ask you if I were to label axon hillock, it would be in a case where it is unambiguously the largest projection in the cell. I wouldn't I would never pick this one to label. Is this the axon hillock? Well, I don't know. What about that one? Maybe this one. Who knows? It's multipolar though. In a multipolar neuron, it can sometimes be difficult to distinguish the dendrites from the axon. All right. So here is a medullated nerve, a myelinated nerve. So what we're doing here is simply looking at, and let's be clear in our language here. When I say nerve, I'm talking about a collection of neurons running along a track, right? So neuron and nerve are two different things. One is made of the other, just like muscle fiber and muscle are two different things. Yeah?
So earlier we talked about the equation of diameter.
Yeah. Mm-hmm.
Is there a way to tell multipolar neurons? Is that something looking at the slide?
So the difference between the various classes of neuron is morphologic, and the morphology translating into functional distinctions. In terms of like diameter, it's really shape. It's really shape that you'll see when we get there. We're not in lab. I'm not going to exhaustively show you slides of all of the different types of neurons. We're just looking at multipolars. But in lecture, you'll see some different shapes. I don't think diameter is probably the right metric. You'll see when you see the different shapes. All right, so in these myelinated nerves or medullated nerves, medulla referring to the inside of something, really just that. The axon, the business portion of these is kind of hiding in this sheath of myelin, right? So the business is the medulla of this structure. They call it medullated. So here's an axon, here's a node of Ranvier, this is all internode, node of Ranvier, and right down the middle you can see a little bit of the excitable membrane. So you'll be able to find, yeah? Yeah, so In this, I would guess that perhaps, so this nerve has been teased apart, first of all. So the connective tissue that would normally be there has been disrupted to a certain extent. But endoneurium perhaps is this outer sort of transparent sheath that you see here. And then the neurolemma would be the outer membrane just below that. And then perhaps this is the axolemma there. Does that make sense?
Yes.
The endoneurium is definitely covering the myelin. Absolutely. Yeah, and you can see it doing that in that model as well. I'm unlikely to ask it here because, I mean, this is a lovely picture, but it would be hard to visualize it that clearly on the slide, probably. If I was going to put this slide up, and if I was a student, maybe here's, when you're studying for this stuff, imagine being the teacher. What is the teacher likely to ask? If I put a slide like this up, what am I likely to ask on this? Well, I'm not cruel. I guess there are some cruel teachers out there, but I'm not. And I'm going to ask you something that's fairly clear, the thing that we have here is the node of Ranvier. That's the clearest thing that you would ask, right? Or maybe I would ask, what is this? This is myelin sheath, or maybe internode. Depends on how I word the question, right? All right, so here's a transect of an actual nerve. Again, you'll find these in slides over here. This is a peripheral nerve somewhere in your body. And what do you notice when you look at this? Looks a lot like a muscle. It really does look a lot like a muscle. I told you earlier that there were going to be some real analogies between the two. Well, and the terminology is quite analogous. It's quite analogous. So around the whole nerve, there's going to be and just replace the word mesium with neurium, and then pretty much all the other language is the same. So instead of epimesium, there's going to be epineurium surrounding a sheath over the entire nerve. Then there are going to be fascicles. The nerve itself will be broken into fascicles. Fascicles being collections of individual neurons. A fascicle is surrounded by perineurium. It's another connective tissue layer. And then within an individual fascicle, each of the neurons is going to be surrounded by endoneurium. Does that make sense? So pretty straightforward transferable terminology from muscle to nerves. All right. And likewise, you can see that the perineurium is the conduit for a lot of all these little spots over here. What is that? What do you guess these holes are? You had to guess. What? Blood vessels. Yeah, the smaller, denser ones being arterioles, the more larger, flattened out, thin ones being venules. And then perforating tiny capillaries are going to be running throughout the actual fascicles. All right, so same slide, just a little bit closer. And here we can see individual neurons. So let's just look at this one, for example. The dark staining area in the middle of that is going to be the axon. And the white surrounding it is going to be the myelin sheath because it's white because it's fat. It's just lipid membrane. That's all it is. Just lipid bilayer, stacks of lipid bilayer. So very hypodense appearing on the image here. Just like in, did I show you a picture? It might have been one of the things that hit the cutting room floor, but did I show you a picture of adipocytes, fat tissues? I did, yeah. So, and the inside of the adipocyte is all white. And the same thing here. So that white ring is simply the myelin sheath. Okay. And yeah, just really getting close in, you can see. So this would be axon, myelin sheath, and then this would be endo. Outside here, all of this stuff would be endo. Maybe capillaries, like that. All right. Okay. So these peripheral nerves, we're working our way up the chain here. So I started with the microscopic anatomy of a nerve, or neuron, pardon me. Then I looked at peripheral nerves, and peripheral nerves are converging on the central nervous system. So we're now transitioning from peripheral to central. And this is the spinal cord, the center, the lowest portion of the central nervous system. The central nervous system, central nervous system in general is divided into gray and white matter, as is the spinal cord here. It has gray and white matter. I really wish this was a whiteboard because I want to draw on this really bad. Because I don't know if, let's see. Yeah, this is a little bit better, but it's still pretty small. Anyways, this would be the model depicting what I'm about to talk about. Let's see. This, bring this down. Should be able to draw on. All right. So first off, there's this thing called the central canal. It runs down the very center of the spinal cord and communicates with what are called the ventricles of the brain, void spaces there. Here is the gray matter. Something like that. Okay. So the gray matter is this darkly staining stuff right here. And within the gray matter, there are three horns. There are gray horns. There's a posterior gray horn. There is a lateral gray horn. And there is an anterior gray horn. All right? There are, and these horns are going to be the place where the cell bodies of various neurons reside. They're going to be kind of like nuclei where the cell bodies exist. And then there's going to be the white matter. And the white matter is divided rather than in horns, we call them columns. In column, the columns suggest, like if you look out here at Miller through the window, a column obviously is something that's going up and down. And that's what is happening in these columns. The columns are white because, does anybody want to guess why columns might be white and the gray may be gray, depending upon, as an extension of what I've already said. Yeah, myelin. So what does that suggest about these columns? This is where there are axons, right? Axons traveling up or down the spinal column are right here. So the sort of longitudinal fibers going up or down the column. So there's going to be a lateral white column, there's going to be a posterior white column, and there's going to be an anterior white column. All right? Now, in the front, the anterior portion, which is, if you're thinking about the spinal column, is actually deep, right? So outside world, the nearest exit is that way, posterior, right? That's the ridge that you can feel. This is anterior. There is this anterior median fissure that comes down this way and that. So the tissue on either side does not communicate. It does not communicate. It does on the posterior side, right? And there's just a little posterior median sulcus. A sulcus is just a little divot, furrow, S-U-L-C-U-S. Here's the word. Posterior median sulcus, S-U-L-C-U-S. Sulcus is a divot. There's a posterior median sulcus, but an anterior median fissure. So the word commissure, C-O-M-M-I-S-S-U-R-E, commissure, is a place where decussation takes place. Let me write that word down. We'll talk about it in lecture. Decussation. For something to decuss, it's just going to go from the left to the right or the right to the left. It's just going to cross the midline. A fiber of some sort, a neuron. It's going to go from the left to the right or the right to the left. And these happen through these commissures. So there's going to be a posterior gray commissure, an anterior gray commissure, and an anterior white commissure. But no posterior white commissure because there's no fissure. I mean, the whole thing is a commissure. They can go back and forth. We don't call it a commissure. All right? The anatomy of the spinal cord. Is there anything else I put on there? Nope, that's it all. Yeah. So I literally, you watched me put this slide in the slideshow, so I didn't get a chance to put any labels up on here. But this is me trying to take models, point stuff out on models more to heart. But there are some things that I can point out. In fact, all of this is the anatomy that I just, right, the cross-section of the spinal cord. So that's all there. There's some other things. This is the dorsal side and the ventral side, right? Posterior and anterior. This nerve, this is a spinal nerve root that's coming out. And there's a dorsal root and a ventral root coming off the posterior gray horn and the anterior gray horn. A dorsal root and a ventral root. The dorsal root, and we'll talk about the functionality of this in class, but the dorsal root has a dorsal root ganglion on it. A ganglion is a ball of neuron cell bodies. It's a place where there's synapse. There's synapse in the dorsal root ganglion. There's a synapse. Then, after the dorsal root ganglion, the dorsal root and the ventral root come together and form a spinal nerve. Spinal nerve. So what's the number for spinal nerve on there, Chris or Liam? It's like 20. 20, yeah. So 20 is the dorsal, the spinal nerve. There's a bunch of other stuff. There's these ligaments that hold it in place. Meninges. So yeah, okay, meninges do that. So in a bit, I'll talk about the cranial meninges, but there are spinal meninges as well. There's 3 layers of meninges. Yeah. So here's anterior. Yeah. Oh, sorry. This is posterior white column. Here's lateral white column. Here's anterior white column. And the roots, the nerve roots, come out of the points of the posterior and the anterior gray horns. And then they come, these are the ventral and dorsal nerve roots. The dorsal root has a ganglion right here. And then after that ganglion, the synapse that happens there, they join into a single spinal nerve. What you will find is that the distinction between dorsal and ventral roots is the distinction between afferent and efferent nerves. And we'll learn what that even means for those of you who don't know in lecture today. All right, on to the brain. So the two most obvious portions of the brain are the cerebrum and the cerebellum. The cerebrum is the big cauliflower thing sticking on top that most people think of when they think of brain. This is what's going to give you your conscious thought, all the supposedly higher order aspects of cognition. This is where the bulk of our memory storage is. Any kind of conscious or subconscious, not fully unconscious, but subconscious regulation happens here of skeletal muscle control happens in the cerebrum. The cerebellum is back here, and it often gets short shrift, but it's an extremely complicated organ that is going to coordinate complex motor functions. So when you're learning to ride a bicycle, you're doing it with your cerebrum. Once you have learned to ride a bicycle, it is the cerebellum that is doing that for you. Right? It's the cerebellum that is doing that. It's coordinating those muscles. So it takes muscle patterns that are trying to be learned and encodes those muscle pattern firings and coordinates the cerebellum. So the cerebellum really is sort of running a lot of the other parts of the brain, makes certain things automatic that are better for us to be automatic. Let's look at the cerebrum in a lateral view and see some of the regions. So it's broken up into lobes. There are 5 lobes. There is the frontal lobe that is. The lobes more or less are named in conjunction with the bones that lay on top of them. The frontal lobe, the parietal lobe, the occipital lobe, the temporal lobe, and there's one other called the insula or insula of Rayl that's hiding in here. But we'll get to that in a moment. So frontal lobe, parietal, occipital, temporal lobes. When you look at the brain, you see it's this wrinkled structure. The wrinkled ridges, the crests of the ridges are called gyri, or in singular gyrus. All right. So the ridge is called a gyrus, and in between, I'd already used the word sulcus. It means just a divot or a furrow. There are a couple sulcuses. So most brains have kind of unique patterns of sulci, although there are patterns, just like your palm has unique patterns of lines, but there are some lines in your palm that everybody more or less has, right? And the same thing here. Everybody has a lateral sulcus that separates the temporal lobe from the frontal and parietal lobes. And then important one is what's called the central sulcus. Central sulcus. And the central sulcus, hand me a brain. The central sulcus has been drawn in pen on this. Please also never use models. This is my brain. A little worse for the wear. The central sulcus separates the frontal and the parietal lobes, but it's an important boundary because it's on either side of it are the precentral and postcentral gyri. All right, the precentral gyrus and the postcentral gyrus. These two ridges of brain tissue are important because they control the somatosensory. They're basically the somatosensory cortex. I'll talk about them in lecture today. All right. There's all the other parts of the brain are important too, but these are very fundamental ones that I'm going to talk about in lecture. So there's the precentral gyrus and the postcentral gyrus, and we'll see what that even means in a bit. And then all of this is sitting on the brainstem. When you're just looking at the brainstem in this undissected view, you see the pons sitting on top here, and the medulla oblongata transitioning into the spinal cord. If you were to pull back to retract the temporal and frontal lobes and look, there is more of the same under there, this sort of hidden, this surprise cortex called the insula of R-E-I-L, rail, insula of rail, R-E-I-L, or just insula, it's commonly called. We'll see what the insula does in a bit, but the other half of my brain comes apart, and you can see the insula right here. Open this up, see the insula model right there. And it's laying on top of the basal ganglia. We'll get to what the basal ganglia are. Okay, so here is a transection down the midline. And if we cut the brain in half and we look down the middle, we're going to see on this model and in this model, we see this like dense white area. This tissue, there is the median sulcus that divides the two hemispheres of the brain. But connecting them is this dense white body called the corpus callosum. Does anybody want to venture a guess as to what corpus callosum means? So if I were to take the off of the end, what is that word? How would you pronounce that? Callus? It's related to the word callus, our word callus. It just means the hard body, the dense body, the calloused, the hard body. So this is an area of extremely dense white matter, which is how the right and left hemispheres connect with one another. Has anyone heard of Oliver Sacks before? The guy the writer, yeah. Have you read any of those books?
Yeah, the man who
mistook his wife for a hat. Yeah, lovely book. Popular science writer, neurologist, talks about various cases. And in the book that she mentions, Amanda mentions, there's a case of a person who had a lesion in the corpus callosum, and the two halves of his brain were not communicating with one another. And the gentleman fundamentally did not recognize the left side of his body as being part of his body. So right hand, right arm, all the stuff over here was part of his body, but he didn't recognize his left arm and leg as belonging to him. So pretty interesting book to read. It's easy to read, very easy to read, entertaining. Oliver Sacks. Anyways, corpus callosum. There is this thing called the fornix right here. And it's kind of hard to show what the fornix is. You can see a little bit of it right here beneath. So looking at the transection, the uppermost structure that you're going to see here is the corpus callosum. Then there's this sort of hollow area which is filled with something called the septum pellucidum, the pellucid septum or pellucid membrane. This is going to be the area that separates the two ventricles. That model there is. We'll get to that in a bit. And then below the septum pellucidum is the fornix. This is what the fornix looks like in isolation, if I were to take it out. There's a small portion that gets transected down the midline. The fornix connects the mammillary body with the amygdala and the hippocampus. Hippocampus is the part of the brain that helps you store. It converts short-term to long-term memory. But fornix is another part where there's decussation between the two hemispheres of the brain and connection between mammillary body. What does fornix remind you of? The word. Yeah, say it. To fornicate. And so to fornicate is to bring two bodies together. All right, is to bring two bodies together in conjugal union. And in the same way, fornix is bringing the two halves together. Fornix. I don't know if that helps you remember the name a little bit. Okay, so here is a picture of the basal nuclei, and I'm showing you these two planes because the next two slides are going to be these cuts. You're going to see these cuts in these slides. The basal nuclei are the caudate nucleus, the putamen, and then the thalamus is sitting beneath it. There's also this thing called the capsule, which is not labeled or not depicted. But the caudate, lateral, the amygdala, the putamen, the capsule. The amygdala is also not labeled here. I showed the amygdala. So we'll see. The acronym that I use is CAPS: caudate, amygdala, putamen, CAP, CAP. All right. So here is the transverse section. And we have the head of the caudate most medially. You can see here fornix. This would be part of the septum pellucidum. Then there's this, so it goes caudate, internal capsule, putamen. On the medial side is the thalamus. The thalamus is part of the diencephalon. There's the thalamus and the hypothalamus. You see better picture. And then I will talk about the thalamus when I get a little bit. But the fornix lays right on top of the thalamus. What else can we see here? We see the pineal gland. Posterior is pineal. Posterior pineal. Pineal, midline here, septum pellucidum. Thalamus. Next in is the caudate, internal capsule, and putamen. You can't see the amygdala because it's deep, it's cut, but the amygdala fits in there. We'll see that in a moment. What else do I want to point out here? So you can see all these void spaces here. All these void spaces are part of the ventricles. So this gray model, Meryl, would you hold that up, please? No. The gray model between you and Maria. Yeah, pull that up. That model there is a model of void space. That is empty space. So where there's plastic, that is actually hollow space. It would be as if you had filled the hollow space in the brain with plastic, and you would get that mold. All right. And that's the shape. It's a little bit complex how it all packs together. But this is part of it here, and you have the septum pellucidum. We'll see a better picture of the thalamus in a bit. There's these two sort of kiwi-shaped lobes here that's connected down the middle through the massa intermedia. We'll see that in a bit. But that, if you look at the ventricles, again, I'll show you. If you look at it, it looks like a moose to me. It looks like a moose with some big horns, giant horns. Anybody ever read the kids' book, like little children's book by a guy named Bill Peet? It was something, what was his name? Somebody, the big horned sheep. It was this sheep that had these enormous horns that he used as skis to escape the hunters. Anyways, you can see the eye of the moose right here. This is where the massa intermedia between the two thalamuses, the thalamus and the other basal nuclei, would sit right in this space right here where my fingers are. Okay. So here is, I guess I have entirely cut the amygdala from. I guess it's not a part of what I thought was necessary. But if we look, here's the putamen, here's the caudate. This is the A for CAPS, C-A-P, the cap, the caudate, amygdala, putamen. Here's the internal capsule radiating out. We can see the insula of Rayl that lays right on top of the basal nuclei. Here's the septum pellucidum. Here's some of the void space from the two ventricles. Yeah. More pictures of the same. Kind of complicated to put it all together. But the best is to just, rather than complicate it with this sophisticated model, or picture, I would say spend some time with the brain and see what it actually looks like on the models. So, anterior, posterior, here's the pineal gland and the pituitary. Pineal gland is the pretty much the third eye. So the pineal gland has photoreceptors, and in lizards, like iguanas, anybody into lizards around here play with an iguana before? No. So lizards don't really have all this stuff, and their pineal gland is actually a pineal eye that can be visualized on the surface of the back of their head. That's non-image forming. If you look at a lizard, they have their third eye right on the back of their head, and it regulates circadian rhythms. So sleep cycles, reproductive cycles, feeding cycles, right on the back of their head. The pineal gland. I could go on, but I actually wanted to stop talking. We can see here the central canal of the spinal cord communicating with the space that is the ventricles. So the central canal of the spinal cord would descend from the tail of these ventricles down this way. All right, we have the two horns of the right and left lateral horns of the ventricles there. Here's the thalamus. So it goes corpus callosum, septum pellucidum, fornix, thalamus. That's the massa intermedia. We'll see it in a moment. And then the next thing is the pineal gland posteriorly or anteriorly. We have thalamus and hypothalamus. It's this triangular region which descends into the pituitary or hypophysis. If you were to look at the bottom of a brain, so remember I talked about the Turk's saddle, that Turk's saddle in the sphenoid bone? The hypophysis or pituitary is it's the Turk that's riding in the Turk's saddle. If you look at the bottom of the brain, so here it is, I'm going to look at it, and it's this sort of little dingleberry hanging off the bottom of the brain that is surrounded by the sella turcica of the sphenoid bone. And the hypothalamus feeds right into it. If you're looking at that, you'll notice, so if the hypophysis is the head, then right below the head are the mammillary bodies, the breasts of the little figure that we're imagining we see. It's a lonely thing to be an anatomist back in the day. This would be the head. This bump right here is called the mammillary body. There's two of them, one on either side. You may remember that the mammillary bodies were at the terminus of the fornix. So the fornix is going to wrap around the hypothalamus and terminate at one of the mammillary bodies anteriorly, and it's going to go back to the hippocampus and the amygdala on the back end. The other structure here is the corpora quadrigemina, the superior and inferior colliculi. Corpora, body, corpora, quadrigemina, the quadruplets, quadrigemina. So pineal gland posterior, below them is the corpora quadrigemina. You'll be able to see those on the back of the brainstem if you open it up. There's some of these brains have better views than others. And then anteriorly is the pituitary and the mammillary bodies. So your face and your breasts are on the front of your body, and pineal gland is very deep. It's like the pineal eye that doesn't see the back of the lizard with the corpora quadrigemina. 4 twins of the brainstem. All right, there's a lot of anatomy there. Here's the thalamus. Here's the thalamus with the massa intermedia connecting them, passing through the eye of the moose. And laying on top of it is the fornix. So this path that you see laid out on top of the thalamus is the fornix. All right. Diencephalon is the name that we give the thalamus and the hypothalamus. So here's half of the brainstem. Here's thalamus on this side. Hypothalamus is hidden by this structure, which we call the cerebral peduncle. Peduncle meaning pedestal or footing, but it's been cut. This is the tissue tract that connects to the big cauliflower brain on top. But if you were to remove that, you would see the hypothalamus here connecting to the pituitary. Below that is so the cerebral peduncle is what's the midbrain. It's part of the midbrain. So diencephalon is thalamus, hypothalamus. Midbrain is essentially the cerebral peduncle. Then we go into the pons. It's the next region down. Then the medulla oblongata. All right. So all of this makes up the brainstem: medulla, pons, midbrain, diencephalon. Who's feeling shocked and appalled by the brain right now? No one? A little bit? Is it blowing your hair back? There's a lot. All right. I'm not even going to pretend to hold you guys accountable for the cranial nerves. We'll talk about them in lecture. There's this stuff called choroid plexus. It is the pink that's painted on this model. And some of the other models may have it painted on there. I don't know which ones. You have to look around. But choroid is in bright red in this picture here. And choroid plexus is tissue that is exposed to the interior of the ventricles, the hollow spaces in your head. And it is what makes cerebrospinal fluid. So the ventricles, the central canal, and then the entire external surface of the brain and spinal cord are all bathed in this fluid called cerebrospinal fluid. It's what holds the nutrients for the brain. There's this thing called the blood-brain barrier we'll talk about in a little bit in lecture. Your neurons are not in direct contact with the blood. Instead, they are in the nutrients and the different chemicals they need are in exchange with the cerebrospinal fluid. And it is the choroid that produces cerebrospinal fluid that gets then modified by contact the fenestrated epithelium surrounding the capillaries. So choroid plexus. Here we see the pineal gland. So this is a posterior view. Pineal gland with the corpora quadrigemina. The superior, the left and right superior, and left and right inferior colliculi that represent the corpora quadrigemina. We will learn about, I'm pointing them out because when I talk about the eye, a lot of the visual processing happens in some integrative processing happens in superior colliculi for sight and for the inferior colliculi for sound, for hearing. There's coordination with, like, you see something flying at you, and there are relays that happen there that enable you to, like, duck. Or you hear something and it makes you duck. Those kinds of signaling events happen in corpora quadrigemina. So I talked about the cerebral peduncle of the midbrain. This is the I took the labeling off, but this is the cerebellar peduncles. The cerebellum would be blossoming off the back of the brain towards you right there. And then the medulla oblongata in the bottom. I'm not going to... Am I going to label any of the cranial nerves? I don't think so. There's a couple that you should know, but these aren't good depictions of them. I mean, you got to know the optic nerves, cranial nerve 2, and then there's a few of them, but I'm not going to go through them right now. Yeah, so here's the front, the anterior of the brainstem. So this is like that little character that I talked about. The hypophysis has been cut off, so the head of the character has been removed. Here are the mammillary bodies. That would be like the torso of that little character. Wrapped around that character's neck, and like a scarf, would be the optic nerves coming to form what's called the optic chiasm that go into the optic tracts that descend on either side. And we will have a discussion about that on Wednesday in lecture. So diencephalon, midbrain, pons, medulla. Okay. Cerebellum. Cerebellum down the midline has this structure called the vermis. Vermis is related to the word. What does the word vermis mean to you? You tell me. What do you think? Huh? Worm. It's a worm. The vermin. The vermis. Vermiform. It's the worm that, and you can see why they would say that runs down the middle of the cerebellum. And then there are these ridges, which we call folia. Folia. Why do you think they call them folia? What does that word mean? Leaf. Yeah, it means leaf. And we'll see why. If you look at a hemisection of the cerebellum, we have white and gray matter. And the white matter, it forms this kind of like tree-like depiction, which the anatomist called Arbor vitae, the tree of life. The tree of life. Tree of life. I used to know this architect named Rich Ahern. He's a pretty interesting guy. I lived in Ann Arbor. He ran a loft called Arbor Vitae Loft. The only reason I'm thinking about him now, besides Arbor Vitae, is that he had brain cancer, and he sent his brain cancer into remission by, I don't even know. This is one of those weird anecdotal stories. He ate pretty much nothing but garlic for a year. Somehow sent his brain cancer into remission. He was a character. Show up to these 75-year-old guys show up to these punk shows in basements, jumping around, pogoing. Just brain cancer. Amazing guy. Arbor Vitae means the tree of life. This guy embodied the tree of life. Somehow beat brain cancer. Didn't beat it. Eventually got him. Came back. But he was old by that point. And the folia are the leaves on the tree, on the Arbor Vitae. So these ridges are called folia. All right. So I haven't talked too much about the connective tissue structure that supports the brain. There are 3 layers of it. They're called dura mater, arachnoid mater, and pia mater. The durable mother, the spidery mother, and pia mater, the small mother. So dura, arachnoid, and pia mater. We'll talk about those in the lecture. But there's this thing called the falx cerebri, and there's another one called the falx cerebelli, and then the tentorium cerebelli. So falx cerebri is this dura, this layer of cranial meninges, this dural layer. Dura is tough. It's the stiff dura that is going to separate the two hemispheres. Laying right here inside this ridge would be what? What brain structure would sit right here? You guys remember? The corpus callosum. Yep. Yeah, callosum. That's right. C-A-L-L-O-S-U-M. Callosum. The corpus callosum would be right here. And this would be the falx cerebri. Kind of like an inverted dura mater mohawk. All right? It just runs down the midline. And it continues for the cerebellum right here called the falx, F-A-L-X, falx. F-A-L-X, falx. What a great Scrabble word that is. What a great Scrabble word. Yeah, it runs. And what would the anatomical feature be that would be right here? I just said it. Say it.
Cerebellum.
The cerebellum. What part of the cerebellum? Say it again? The vermis. Yeah, good job. Good job. And then separating the cerebellum and the cerebrum and the cerebellum is this tentorium. T-E-N-T-O-R-I-U-M, cerebelli. Tentorium cerebelli. Tentorium, related to the word what? My brain isn't pulling it up, but there's an English word that it's related to, but basically meaning interrupting or separating. It's separating the two structures. In this falx cerebri, in all of these, there's all kinds of vasculature. The only one I'm really going to talk about is simply the superior sagittal sinus. I'm going to talk about it when I talk about in lecture how cerebrospinal fluid is recirculated in the brain. I need to mention the superior sagittal sinus. So this is what it is. There's a sinus. It's a venous structure that's draining the brain, running up along the superior margin of the falx cerebri. Here's a great picture, just as by and by. I showed you the brainstem where it had the pituitary gland cut off, and you can see the mammillary bodies. Whatever. But here it is, sitting in the sella turcica of the sphenoid bone. Here's the sphenoidal sinus from the lab yesterday. When you have hypophyseal tumors, adenocarcinoma of the hypophysis, adenohypophyseal tumors, the way you operate on it, you can't like take the brain out of the head and deal with it and put it back. They actually deal with it by breaking the bone in the sinus and doing brain surgery through your nose. Pretty amazing thing, man. Pretty amazing thing. Here's the ventricles. So a little moose. There is the two lateral ventricles, left and right. Then there's the third ventricle, and it has this little hole there, the eyepiece of the moose, which is where the massa intermedia of the two thalmuses connect. Goes down through the cerebral aqueduct down to this fourth ventricle, and then finally into the central canal of the spinal cord. So those are the ventricles of the brain, the holes in the head. And I think, so here's a different view of them. I think that is all I'm going to say.